all see my screen here?
Yep.
All right, perfect. Thank you so much, Dr. Jane, for the introduction, and thank you all very much for being here today for my talk. So like he just said, I'm going to be talking about a method that I published in communications biology earlier this year called SpaDecon. And this is a method that we developed to perform cell type deconvolution in spatial transcriptomics. And it's based on a semi-supervised learning framework. Also, I'm happy to take any questions throughout the talk as well. So if you have any questions, yeah, please just ask, and I'd be happy to stop and go back and answer those. But I'm just going to start the talk with going over a brief description of the evolution of different transcriptomics technologies over the last decade. So in 2013, single-cell RNA sequencing was named Method of the Year by Nature Methods. And this method was really revolutionary because it was the first of its kind to really be able to profile gene expression within individual cells in a tissue section. Then later in 2019, single-cell multimodal omics built even further upon single-cell RNA sequencing and allowed for the profiling of multiple modalities within individual cells in a tissue section. And while both of these technologies were extremely revolutionary for disease analysis and treatment purposes, one major downfall to both of them is that they lack spatial location information of cells because the cells need to be dissociated from tissues prior to being sequenced. And this is extremely crucial because the knowledge of the spatial location of specific cell types is extremely necessary for disease analysis and treatment purposes. And this all kind of started to change with the development of spatially resolved transcriptomics, which was named Method of the Year by Nature Methods in 2020. And this technology was really the first of its kind to give that spatial context to gene expression and allow for an understanding of gene expression within a spatial context. And there are many different types of spatially resolved transcriptomics technologies, but all of them measure gene expression at some predefined spatial locations, whether it be individual cells or spots containing multiple cells. And many of them also provide some morphological context to the spots in which gene expression are measured as well. So next, I'm just going to give an overview of the two main types of spatially resolved transcriptomics technologies. The first are imaging-based SRT technologies, which use fluorescent probes to localize genes at precise locations with subcellular resolution. So the resolution of these technologies is higher than that of any others. It's able to find pretty much the exact spatial location in which a specific gene is expressed. So the resolution is extremely high. The main downside to these technologies, however, is that the gene panel must be pre-selected, and you're limited to identifying at most around 10,000 genes. So it's nowhere near like full transcriptome level. And 10,000 is really a bit of an extreme case. In most cases, you're really only limited to identifying about 500 genes before they start to overlap with one another, and the signal isn't as good. Examples of these technologies include MERFISH, which is now commercialized under the MERScope technology, SeqFish Plus, and Xenium, which is commercialized by 10x. And along with the spatially resolved gene expression data, a lot of these technologies also provide DAPI images, which give morphological context to the cell nuclei. Okay. And the other main type of SRT technology, which is the one that I'll be focusing on for my talk because it's what we run SpaDecon on, is sequencing-based SRT technologies. And unlike the imaging-based technologies, these don't provide data with single-cell resolution. These measure gene expression at capture locations referred to as spots, which can contain multiple cells. And the size of the spots and the number of cells per spot varies greatly on the technology that is used and the tissue section which is being analyzed. For 10x Visium, which is one of the newest sequencing-based SRT technologies, the spot size is approximately 55 microns in diameter, and the number of cells is typically between 1 to 10 per spot on average. Spatial transcriptomics, however, is a much older technology, and the spot size is a lot larger, and some tissue sections contain up to 200 cells per spot on average, so nowhere near single cell resolution. The benefit to these technologies, however, is that they can measure gene expression levels for the entire transcriptome, and you're not restricted to pre-selecting a few hundred genes to measure expression for. And this slide just kind of shows the difference between two different sequencing-based SRT technologies. So on the left is a tissue section that was analyzed using 10x Visium from a breast cancer tissue section. And on the right is a melanoma tissue section analyzed using spatial transcriptomics. And overlaid on the histology, I have just kind of the relative size and locations of the SRT spots that were sequenced. So you can see for the Visium data set, the spots are a lot smaller and a lot closer together compared to the spatial transcriptomics data set. And there's also just a lot more of them. So in this Visium data set, there's approximately, I think, like 3,500 spots compared to the ST data set, which only has a few hundred spots. So Visium really improved upon the resolution of that was present in spatial transcriptomics. And these are the two types of data that I analyzed SpaDecon on and that I'll be showing results for today. So as I mentioned, the problem with like single cell RNA sequencing and single cell multimodal omics is that they lack any sort of spatial information, which is problematic because understanding cell type distributions is crucial for disease analysis. And in sequencing-based SRT technologies, even though we have that spatial information, genes are measured at spots containing multiple cells, which hinders inference at the cell level. So we can't just use a typical like single cell clustering algorithm or cell type annotation algorithm to assign each spot to a specific cell, because each spot can contain multiple cells of different types. In this case, however, when we have multiple cells per spot, cell type distributions are often estimated by inferring cell type proportions at the spot level. And this is done via a cell type deconvolution algorithm. We also know that gene expression, spatial location, and histology information are all informative for localizing cell types. And histology information can provide a morphological context to cells within spots that aren't necessarily known with just gene expression or spatial location information alone. And the problem with cell type deconvolution methods that were developed before SpaDecon is that they don't account for the spatial and histological dependence of spots, but they rely pretty much entirely on gene expression in order to perform cell type deconvolution. And this is just an overview of some of the methods that were developed before SpaDecon, and they're the ones that I'll be comparing with for the results section of this talk. The first method is robust cell type decomposition, or RCTD, which was published in Nature Biotech a few years ago. And this is a maximum likelihood-based approach for estimating cell type proportions, and it assumes that gene counts follow a Poisson distribution. Next, there's Spotlight, which was published in Nucleic Acids Research, which is a seeded non-negative matrix factorization regression based framework. Then Stereoscope was published, I think in 2019 in Communications Biology, and it's a maximum likelihood estimation approach for cell type proportion estimation, assuming gene counts follow a negative binomial distribution. And then finally, Subtolocation, published in Nature Biotech, is based on a hierarchical Bayesian framework, assuming gene counts are negative binomial distributed. And as I mentioned, the main downside to each of these methods is that they don't account for the spatial and histological dependence of spots when inferring cell type proportions. And that's what motivated us to develop SpaDecon, which is a semi-supervised learning method for cell type deconvolution in spatially resolved transcriptomics that utilizes the SRT gene expression matrix, spot spatial locations, and the high resolution hematoxylin and ISN histology image accompanying the Visium and ST data. SpaDecon also requires an annotated scRNA-seq reference dataset, which is a gene expression matrix that has cell type labels for each cell that is sequenced. All right, and next I'm just going to provide a brief overview of the SpaDecon algorithm before I go into the details of each of these steps. So the first step is the step in which we're actually incorporating the spot spatial locations and histology image. And this is in order to perform a local smoothing of the SRT gene expression matrix. The second step is just pre-processing of scRNA-seq and SRT gene expression data using just standard pre-processing steps. And the third and fourth parts are the actual deep learning component of SpaDecon, which comprise the semi-supervised framework. So step 3 is an unsupervised training of a stacked autoencoder in order to find a mapping to a lower dimensional space for our very high dimensional gene expression data. This is followed by a supervised clustering algorithm in order to optimize model parameters so that the network can find features that are relevant for cell type classification. And then finally, we do inference of SRT cell type proportions by inputting our SRT gene expression data into our trained model. And like I said, I'm going to go into detail on each of these steps. So that first step is local smoothing of the SRT gene expression matrix using the spot spatial locations and histology image. So what we're looking to do is construct an adjacency matrix that represents the similarity between each pair of spots in our SRT data set with respect to spatial location and histology. And the similarity metric that we're going to use is based on the Euclidean distance with respect to spatial location and histology. So calculating the Euclidean distance between each pair of spots is very straightforward with respect to spatial location. We just take the square root of the sum of the squared differences between the x and y spatial coordinates between two spots. However, for histology, this isn't as straightforward because we don't have a single value representing histology for each spot in the data set. So we need to actually construct that value for by ourselves. So that value for spot V is given by ZV here. And it's just this weighted sum of the means of the RGB values across all pixels in the 50 by 50 pixel square centered on that spot. So what we do is for each spot in the data set, we draw a 50 by 50 pixel spot box surrounding that spot. And for each of these pixels, we take their RGB values and we average over them in order to get the mean of the RGB values for all pixels centered on that spot. We then weight each of these mean RGB values by the variances of these RGB values across all spots in the data set. And the reason that we do this is we want to give a higher weight to the channel that has the greatest variance across all spots in the data set, because this channel shows the greatest deviation and therefore will have the most meaning in differentiating different spots based on histology. We then divide by the sum of the variances in order to normalize this value as well. So that's how we get our initial value ZV here. But we also want to put this on the same scale as our x and y spatial coordinates so that when we actually do the local smoothing, the histology will have basically the same influence as the x and y spatial coordinates. So what we first do is do a z-score normalization by subtracting off the mean and standard deviation across all spots. We then multiply by the maximum of the standard deviation of our x and y spatial coordinates so that the standard deviation of our histology value is the same as the maximum of x or y spatial coordinates. We then have this histology scale parameter s, which is by default set equal to 1, so that the influence given to histology is the same as the x and y spatial coordinates. However, we can either increase or decrease this value to provide a higher or lower weight to histology relative to spatial location information. But by default, it's just set equal to 1. If the histology is of bad quality, though, and there are many artifacts, it would be beneficial to lower this value so that less weight is given to histology and more weight is given to spatial location information. All right, and now that we have that value z star, we can then calculate the Euclidean distance very straightforward between spots u and v. And this is just the sum of the square root of the sum of the squared differences between our x, y, and z star coordinates for two spots u and v in our data set. And we do this for all pairs of spots that we have. So our adjacency matrix is quite large. And then we calculate our adjacency matrix by doing a normal distribution kernel for using this distance d at uv, and then divided by this 2 times L squared, where L is a characteristic length scale hyperparameter. And L is selected in SpaDecon automatically based on however much influence the user selects they want to give to neighboring spots relative to a given spot when doing the local smoothing. So by default, L is set so that the influence given to neighboring spots is half of that given to a given spot when calculating the smooth expression for that spot. We then calculate our smooth SRT gene expression matrix through matrix multiplication between our adjacency matrix A and our original SRT gene expression matrix X. So X star is our final smooth SRT gene expression matrix. All right, so that's all for step 1. That's the only place where we're actually using the histology and the spatial location information. It's just to do that smoothing on the SRT gene expression matrix. We then further pre-process our scRNA-seq and SRT data. And this is all pretty straightforward for SRT data analysis. We first do an identification of highly variable genes in the SRT data. This is followed by transcript per million or TPM normalization of both our SRT gene expression data and our scRNA-seq gene expression data. We then filter out all cells, spots, and genes with low counts in both data sets. And we then do a log and z-score normalization of the gene counts so that they're comparable between the data sets and so that they're more suitable for input into our deep learning framework. All right, and that leads us into our actual deep learning framework of SpaDecon, which, as I mentioned, starts with an unsupervised initialization of model parameters. And the entire purpose of this step is just to find a mapping to a lower dimensional space. So since this is unsupervised, we're not using the cell type labels at all for our scRNA-seq data. We're just using our unlabeled SRT data and our scRNA-seq data without the labels. So what we do is we combine our SRT gene expression data, which has been preprocessed, and our preprocessed scRNA-seq gene expression data, and we use those as input for a denoising autoencoder, which is just an autoencoder which sets some of the features randomly equal to 0, so that the model isn't relying too heavily on certain features to reconstruct the data. So we combine those data together, and then for each sample, we input it into this autoencoder, which has a mapping to a lower dimensional space, and then tries to reconstruct the output. And this model is, this network is trained by minimizing the mean squared error between the uncorrupted input before any features are set equal to 0, and then the output of the autoencoder. We further take the encoding of that autoencoder and use it as input for a second denoising autoencoder, which has the same structure, just with lower numbers of nodes for each layer, so that we can reduce the dimensionality even further. But the second denoising autoencoder is again trained by minimizing the mean squared error between the uncorrupted input and the output of the network. And then once that second denoising autoencoder has been trained, we combine the two autoencoders by concatenating the encoding layers in layer-wise training order and concatenating the decoding layers in reverse layer-wise training order, which forms this stacked autoencoder with a bottleneck layer in the middle. And we further train this network by fine-tuning the weights by minimizing the MSE between the original input and the final output. So we have really 3 stages of network training in order to construct this autoencoder.
Kyle, just a quick question on the model. So how deep is your model? So it just says, so from this figure, I can say it's like a 5-layer model.
Yeah, so the number of layers that we have depends on the number of spots and the number of cells in our SRT and scRNA-seq gene expression data. For most of the data that we're working with, it's a total of 2 denoising autoencoders, each with like 3 layers. So 1 input, 1 bottleneck, and then 1 output. So this is kind of the standard structure for most of the data that we work with.
Yeah, sure. Thank you.
Yep, no problem. Okay. And as I mentioned, the entire purpose of the unsupervised portion of the framework is just to find a mapping to a lower dimensional space that's kind of more suitable for our analysis. So once we've fully trained our stacked autoencoder, we can then remove our decoding layers and just keep the encoding layers to have that mapping to a lower dimensional space. And that leads us to the supervised portion of our algorithm, where we're actually going to be using the annotated cell types from our scRNA-seq data set. And that's done by adding on a clustering layer to the final layer of the stacked autoencoder. And I'm next going to go into more detail about really what that clustering layer is doing and what it's trying to optimize for. So at this stage, we're no longer using the SRT data for model training. We're solely using the labeled scRNA-seq data in order to cluster cells in our latent feature space using cell type labels. So the activation function that we have for our clustering layer is given by Qij here, which can be thought of as the similarity between cell I and cell type J in our scRNA-seq data set. So here Zi is the embedding for cell I, and mu J is the centroid for cluster J. So each cluster centroid in this network represents a cell type that is present in our scRNA-seq data set. And the cluster centroids are initialized using the means of the cell type embeddings for each cell type in our data set. So this is based on Euclidean distance between the embedding for cell I and the centroid for the cluster representing cell type J. So again, Qij is just a similarity metric representing the similarity between cell I and type J. And then we also have a target distribution that we're trying to drive these Qijs towards, and that's given by Rij, which is approximately equal to 1 if J is the type of cell I, and approximately 0 if J is not the cell type of cell I. And we can define the target distribution in this way, again, because we are using the labels of our scRNA-seq data set. So what this is doing is it's trying to drive cells closer to the centroid representing their cell type and further away from the centroids of representing cell types that are not of their type. So it's driving cells of the same type closer together and cells of different types further apart. And the objective function that we're looking to minimize is the Kullback-Leibler or KL divergence between these distributions R and Q. And we minimize L by updating the model parameters and cluster centroids through stochastic gradient descent with momentum. And we train a model either until 2000 epochs are reached, or if the KL divergence between two consecutive iterations decreases by less than 0.01. So if we're not seeing a significant difference in the KL divergence between consecutive iterations, then we'll stop the model training. All right, and that's all for the supervised learning portion of the algorithm. So at this point, our model has been fully trained. And once the parameters are optimized, we then input the smooth and pre-processed SRT gene expression data as input for the model. Cell type proportions are then estimated using the distance between each spot's embedding and the cluster centroid for each cell type in the latent feature space. So we again use that same activation function Qij. This time, though, instead of a cell Zi being the embedding for cell I, it's the embedding for spot I in our SRT data set. And instead of representing the similarity between cell I and cell type J, Qij represents the estimated proportion of cell type J in spot I. So we calculate this metric for all spots for all cell types in order to come up with our estimated distribution of cell types throughout all spots in the tissue section. Okay. And that's all for the method section. So I'm happy to take any questions there if there are any, or I can also wait to the end as well if there aren't.
Do you have any question? If not, I have one question. So to be able to use your software, I think the input data basically is spatial transcriptomics data and also single cell RNA-seq reference data. So in terms of the single cell RNA-seq reference data, do we have to have a paired, single cell RNA-seq data, or can we use any public available single cell RNA-seq data from the database? Do you have any suggestion for that?
Yeah, so it can be any publicly available scRNA-seq data set. It's definitely ideal if it's from a paired sample, but that's not very common. Only one of the data sets that I analyzed actually used a paired scRNA-seq data set. All of the other ones were just publicly available from completely different samples. So yeah, it's either one works for sure.
So so basically we can only use one single cell RNA-seq reference data set at one time, right?
Yeah,
can we use multiple one also, in terms of the cell type annotation? So how should we actually choose which single cell RNA-seq data is better, for this project, for example, here?
Yeah, so it's a good question. You can definitely use more than one as well. Like if you normalize them properly, you can combine multiple single cell RNA sequencing data sets, and that'll actually optimize both parts of the network better, both the unsupervised dimension reduction and the actual clustering part. If you have to choose one though, you should choose it based on whatever cell types you're looking to identify. So a lot of annotated single cell RNA sequencing data sets will have different cell type labels. Either some will be more specific than others. And so you should choose whichever one has cell types that you're most interested in identifying in your spatial data set. Yeah,
I see. That makes sense. Thank you. And we have one question in the chat, in the Q&A.
Yeah, let me open that.
How to determine cell types?
Okay. Is that specific for? So the cell types are what are present in the scRNA-seq data set. So the cell type proportions that we're estimating are just whatever are there. Does that answer the question?
Yeah, I'm not sure Biju actually can speak or not. I guess so, yeah.
I see, okay.
Yeah, thanks.
Okay, yeah. Okay, yeah, he said that answers it. All right, cool. All right, so yeah, that was it for the method section. So now I'll move on to some of the results we have from analyzing some real data sets. So the first results I'm going to show are for a mouse anterior brain data set that was analyzed using the 10x Visium technology. So when analyzing real data, it's a bit difficult because there's no ground truth. So we have to go based off of kind of known biological facts about a data set when kind of interpreting the results. So we ran all cell type deconvolution methods that I discussed earlier, including SpaDecon, on this data set, and kind of looked to see how these methods did in identifying cell types within the cortical layers of this tissue section. So in the scRNA-seq data set that we used as a reference, there were 4 cell types that were labeled according to the cortical layers in which they were obtained. So one of those is L2-2-3, which are neurons found in the L2 and 3 layer. Same with L4, 4-5, neurons found in the L4 and 5 layers, and then L5 and L6, neurons found in the L5 and L6 layers. So when we visualize the estimated proportions of these cell types across spots in the data set, we should see kind of the cortical layer structure of the tissue section revealed. We should see high proportions of these neurons in their respective layers and low proportions elsewhere, just like we see for SpaDecon. So we see in this L2 layer at the top, high proportions of this L2-2-3 cell type and low proportions elsewhere. And as we go further to the inside of the cortical layers, we see high proportions of those cell types revealed, and then low proportions elsewhere. When we look at the other methods, however, we see very high proportions of at least one of these cell types in a region where there should not be high proportions. So if you look at RCTD, stereoscope, and cell2 location, they all predict high proportions of L2-2-3 and the L2 and 3 layers, but they also predict very high proportions within the middle of the tissue section, which we really shouldn't be seeing. And then same for L6 and RCTD, it predicts high proportions in pretty much the entire tissue section. And then Spotlight predicts high proportions of L4 and L5 in the middle of the tissue section as well. So SpaDecon by far had the best specificity, it looks like, with localizing these neurons into their respective regions. It predicted high proportions in their respective regions and low proportions elsewhere. So this is very promising for the performance of SpaDecon. All right, and the next data set that I have results for is a breast cancer tissue section that was also analyzed using the 10x Visium platform. So on the top here, I have what are called a scatter pie plots for each of the methods, which it colors each spot as a pie chart according to the estimated proportions of these different cell types in that spot. So each spot has however many colors there are cell types for. And each spot is colored according to the estimated proportion of each of these cell types in that spot. And the main thing to note is that for SpaDecon and Spotlight, you see a lot of different colors, which should be common for this tissue section because it's a cancerous tissue section, which means that it's very heterogeneous. You have a lot of spots that are a combination of cancer cells, immune cells, and cancer-affiliated fibroblasts, and so on. However, when looking at RCTD and cell2 location, a vast majority of the spots are just bright green, which represents cancer epithelial cells. Even in the regions of this tissue section that aren't tumor, they have very high proportions of these cancer epithelial cells. So it does a good job they do a good job in predicting high proportions of cancer epithelial cells within the tumor regions, but also have very high proportions where there really should not be. Whereas SpaDecon did not predict high proportions of cancer epithelial cells outside of the tumor regions. Also, the inferred proportion of cancer epithelial cells within the tumor regions for SpaDecon is a bit lower because it also predicted high proportions of immune infiltrating lymphocytes, which should be expected in a tissue region like this as well. There are also two main differences between SpaDecon and stereoscope, where stereoscope predicted very high proportions of cancer epithelial cells, where SpaDecon did not. And those are labeled as 1 and 2 on the histology image on the right. And when looking at this image in a bit more detail, it's clear that the histology image shows differences in morphology among the cells in these regions 1 and 2 compared to the tumor regions in the tissue section. And the cells are much less densely populated and just show quite a few differences in cell morphology. So it's quite possible that these actually aren't tumor regions at all, and SpaDecon was able to avoid misclassifying them as so because of its use of histology in cell type deconvolution. So this is, again, very promising for our method. Yep.
I think that's very interesting. I think you basically use some like histology data also to have the annotation here. I'm just wondering, like when you compare data, analyze data by use different software here, do you use what's the same single cell RNA-seq reference data also?
Yep, the same single cell reference data is used. And the same pre-processing steps are used. The only difference is that we do the local smoothing of our SRT gene expression matrix, which the other methods do not. Yeah.
I see. Great. Thank you.
Yep, no problem. Okay. And the next data set I'm going to show results for is from a different platform. It's from the spatial transcriptomics platform as opposed to 10x Visium. So if you remember from the beginning of my talk, the resolution of these spots are not as high as they are for Visium. The spots are much larger in diameter, and these spots can contain sometimes up to 200 cells per spot on average. So there's likely going to be even more heterogeneity with respect to the number of cell types per spot. And this specific data set that I'm looking at is from a tissue section with stage 3 cutaneous malignant melanoma. And on the bottom right of this slide, I have a pathologist annotation of this tissue section, where they separate the tissue section into 3 main categories. The first is melanoma, which is circled in black here. The second is lymphoid tissue, which we see in light blue, and then stroma, which is circled in green. And one of the things I wanted to look at here is kind of the localization of malignant cells. So in the scRNA-seq reference data set that I used, one of the cell types is labeled as malignant, and we should be seeing high proportions of this malignant cell type within the melanoma region and low proportions elsewhere. So we can see that just based off of the heat maps of each of the methods, they all predicted high proportions of malignant cells within the melanoma region. But we also see that for a lot of the methods, especially stereoscope and cell 2 location, there are very high proportions of these cells outside of the melanoma region, specifically within the stroma. So what we wanted to do is isolate all spots that were contained within the stroma and look at the distributions of estimated malignant cells within those spots given by each method. So that's exactly what this box plot is showing here. For all spots that are located in the stroma, we're visualizing the distribution of the estimated proportion of malignant cells as estimated by each of the methods. So we can see that SpaDecon on the left by far has the lowest estimated proportion of malignant cells in stromal spots. And stereoscope and cell 2 location have very high estimated proportions of malignant cells in stroma spots. The average proportion of malignant cells estimated by each of these methods was almost 0.5, which is a lot higher than we would expect to see in the stroma region. So this is again evidence that SpaDecon was more accurate in localizing malignant cells into the melanoma region compared to the other methods. All right. And then the last real data set that I'm going to show results for is from a tissue section from a patient with pancreatic ductal adenocarcinoma. And this was again analyzed using the spatial transcriptomics or ST platform. And on the right-hand side, on the bottom right, we have another pathologist annotation for this tissue section, where they separated the tissue into, again, 3 categories. The black region represents cancer cells, the red region is interstitium, and then the green is duct epithelium. So what we would expect to see for this tissue section is high proportions of cancer cells within the cancer region, and again, low proportions elsewhere, which in the scRNA-seq reference data set that we used, there was a cell type labeled as cancer clone A, and this was the only cancer cell type that was present in this data set. So we would expect to see high proportions of this cell type within this black region here. And we can see that the pattern of SpaDecon for the distribution of cancer clone A cells very closely matches where we see the cancer cell rich region annotated by the pathologist. And what we want to do is visualize the distribution of the proportions of these cancer clone A cells within the cancerous spots. So that's the box plot that we have here on the left-hand side. And there's quite a bit of variation between the minimum and the maximum of estimated proportion of cancer cells in these cancerous spots given by SpaDecon. But that's almost to be expected because, as I mentioned earlier, this is an ST data set, so the spots are really large. We can have up to 200 cells per spot. And because the cancer cell region isn't completely contiguous, we would expect there to be kind of a combination of different cell types. But overall, we're seeing a very high proportion of these cancer clone A cells in this cancer cell rich region. And the mean of the SpaDecon estimated proportions of cancer clone A cells in cancer spots was higher than all the other methods, even though the median was slightly lower than RCTD and cell-T location. But this is, again, promising that SpaDecon is accurately localizing cancer clone A cells. Right. And as I mentioned before, that when analyzing real data, it's a bit difficult because there's not like a really good way to come up with quantitative evaluations to compare method performance, since there's no ground truth to compare with. So in order to deal with that, we wanted to construct a pseudo SRT data set that was based off of benchmark cell type proportions, so that we could then analyze that data set and compare with the benchmark proportions to determine model performance. So we constructed the pseudo SRT dataset to resemble the ST pancreatic ductal adenocarcinoma dataset that I described on the last slide. We treated the mean of the SpaDecon, RCTD, Spotlight, and stereoscope answer to location estimated proportions as the benchmark proportions. So for each of these methods, we ran them on the real ST pancreatic ductal adenocarcinoma dataset and took the means of their estimated proportions across all spots in order to construct our benchmark proportions. And the reason we did it this way is we wanted our pseudo SRT dataset to resemble a realistic SRT dataset, but we also didn't want it to be biased towards any one particular method. So by taking the mean, it would help in preventing it from being biased, as opposed to just using one of the methods proportions as the default. We then sampled cells from a paired annotated scRNA-seq dataset according to these benchmark proportions for each SRT spot, and constructed the SRT dataset based on the cells that were sampled by summing over their counts for each spot. We then inferred cell type proportions of the SRT dataset using each method and evaluated model performance using cell type level mean squared error and spot level Jensen-Shannon divergence between the benchmark and the estimated proportions. And Jensen-Shannon divergence is just a distance metric that is based off of the Kullback-Leibler divergence. All right, and here are our benchmark evaluation results when looking at the cell type level MSE between the benchmark and the estimated proportions for each method. So here on the x-axis, we have each of the different methods we used, and we also included MUSIC, which is a cell type deconvolution algorithm that was developed for bulk RNA sequencing, but not really used too often for SRT data analysis. And then on the y-axis, we have MSE. So these are showing the distributions of the MSE across the different cell types for each method. And we can see that the distribution for SpaDecon just visually looks to be much lower than that for each of the other methods, indicating that it is more similar to the benchmark cell type proportions compared to each of the other methods. And then when looking at specific metrics, we see that the mean of the SpaDecon MSE across all cell types is 0.014, which is lower than all the other methods. The second best was actually MUSIC, which was 0.016, which was interesting because, like I mentioned, this wasn't even developed for SRT cell type deconvolution. The median for the SpaDecon MSE was also the lowest at 0.004. The next lowest was Spotlight at 0.008. And the last row shows the number of cell types for which that method's MSE was the minimum across all methods. So basically the number of cell types for which that method outperformed all other methods. And for SpaDecon, that number was 6, compared to 1 for RCTD, 0 for Spotlight, 0 for stereoscope, and then 2 and 4 for cell to location and MUSIC, respectively. So when looking at cell type level MSE, it's clear that SpaDecon outperformed all other methods on this data set. Then this plot shows the results when looking at the spot level Jensen-Shannon divergence between the benchmark and estimated proportions by each method. And we can again see that the distribution for SpaDecon is much lower than all the other methods, indicating closer similarity to the benchmark proportions compared to the other methods. And then looking at specific metrics, we see that the mean of SpaDecon is again the lowest at 0.30. The next lowest is Spotlight at 0.36. And then the median across all spots is 0.28. Next closest is again Spotlight. And then the number of spots for which that method's JSD with the benchmark proportions was the least. Again, for SpaDecon, it outperformed all other methods for 104 spots. and the next closest was Spotlight at 50. So both of these results together are very promising that SpaDecon is quantitatively outperforming the other methods on this data set. And then these next plots just show spot level Janssen-Shannon divergence between the benchmark and estimated proportions for each method. So spots that are white and red indicate close similarity to the benchmark proportions, and spots that are purple, blue, and black indicate a large divergence between the benchmark and estimated proportions. So for SpaDecon, a vast majority of the spots are red and white. There are a few blue spots and purple spots here as well. But overall, in most regions of the tissue section, SpaDecon is performing extremely well. All right, and then this plot just shows the computational runtime of SpaDecon compared to the other methods when evaluated on each of the data sets that I presented today. So we have SpaDecon in red, and then in gray, we have SpaDecon without the local smoothing step. So one of the concerns with SpaDecon is that the first step that we use for incorporating histology and spatial location information is constructing an adjacency matrix, which can be quite large depending on the number of spots in the data set. So if we have 3,000 spots in a typical Visium data set, this matrix will be 3,000 by 3,000. So there's a bit of a concern with this kind of increasing the computational complexity of increasing the computational runtime of our method. However, when comparing just the red and the green, the red and the gray bars, they're really similar across all methods, regardless of the number of spots in the data set, which is promising that the local smoothing step isn't taking up that much time. Also, in parentheses next to each method, we have the number of cell types present in the scRNA-seq data set that we used, and then the number of spots in the SRT data set, just to kind of show how that influences some of the runtime performances. For instance, you can see that RCTD, when looking at the ST data sets like melanoma and the pancreatic ductal adenocarcinoma data set, has a very low runtime. In the case of melanoma, even lower than SpaDecon. However, when the number of spots becomes quite large, like in the mouse anterior brain data set and the breast cancer data set, the runtime of RCTD increases by quite a bit and is usually over double the runtime of SpaDecon. Also, so for Spotlight, the runtime is pretty consistent. You can see there's a slight decrease in runtime when looking at a fewer number of spots. For stereoscope, however, the runtime is extremely long, regardless of the number of spots that are in the data set. And this was using the CPU, but with using the CPU, it still took about a day to finish running for each of the data sets. When looking at cell location, we use the GPU because if we use the CPU to compare with the other methods, it would have taken multiple days, it looked like, to finish. But even with the GPU, it took multiple hours to run because our runtime is in log minutes here on the y-axis. So overall, in terms of computational efficiency, SpaDecon is much faster than most of the other methods, especially if there are a large number of cells in the large number of spots in the data set. All right, and so just to wrap up, I've showed that SpaDecon is a semi-supervised learning-based method that incorporates gene expression, spatial location, and histology information to provide accurate cell type proportion estimates for spots in a SRT data set. I also showed through evaluation on real SRT data sets that the SpaDecon results align well with known biological properties that we know to be true for each of the data sets. Also, SpaDecon outperformed other cell type deconvolution methods when evaluating a pseudo SRT data set constructed using benchmark cell type proportions and resembling a real SRT data set. And then finally, SpaDecon is computationally efficient and analyzed a majority of the data sets faster than all other methods that we evaluated with. All right, and I would just like to thank my advisor, Dr. Mengyao Li, for everything she has done in supervising me throughout the last 4 years. And then our collaborators, Jin Hu, who's a faculty at Emory, and then Edward Lee, who's a pathologist at Penn. And then all of the Laboratory for Statistical and Translational Genomics at Penn as well. And here are my references. So thank you all so much for listening, and I'm happy to take any other questions as well.
Great. Thank you so much for the great talk. Very interesting software. Do we have any question? Please raise your hand or just type the question in the Q&A. Okay. Well, I'm not sure whether we have more question or not, but just wondering, like when you show the computational runtime for different software, yeah, I was just wondering, like for the small data set, like PDAC, you know, dash B, that data set seems like the runtime for your software is like negative value.
Oh, so this is in log minutes. I should have emphasized
Oh,
that.
log, okay.
So yeah, it
makes
was
sense.
about like I think 45 seconds to run. So it was negative in log time.
Okay, that makes sense now. Yeah.
Yeah.
Okay. Do we have more questions? All right. I think that's. I don't think we have more questions from the audience. Well, thank you so much for the great talk. And have a nice day.
Yeah. Thank you again so much for having me. I appreciate it.
Yeah. Great.
All right.
Bye.